Welcome everybody to the second SynBioBeta Town Hall on the new coronavirus. Today it's an honor to be joined by Dr. Charles Chiu at UCSF, the founder of Opentrons, Will Canine, the founder and CEO of Mammoth Biosciences, Trevor Martin, and the head of research and development, Rebecca Nugent at Twist Bioscience in South San Francisco. So welcome everybody. Will, you're normally in New York, but you're joining us live from the Bay Area as you're setting up some new hardware for testing of coronavirus, which we're going to be talking about soon. Trevor, you're joining us from your lab in South San Francisco, and you just published yesterday with Dr. Chiu a brand new paper showing detection of coronavirus using CRISPR. So in 30-minute detection system after you've amplified the RNA from a patient sample, and we're going to be coming onto that. And Rebecca, you just launched this morning a brand new product from Twist, which is actually coronavirus and how it is being used as a reference standards for positive controls. So we have a lot of late breaking news that we're very excited to tell you about. Dr. Chiu is at the epicenter of the brain trust for the coronavirus, specifically here in the Bay Area. And so, Charles, I know we've only got you for 30 minutes, so I wanted to dive in and just ask you if you could just give a brief explanation to say what you do at your clinic and what you're seeing coming in on the ground there at UCSF.
Absolutely. So I have both a clinical laboratory as well as a research laboratory that is focusing, in fact, we've shifted our focus largely to studying COVID-19, which is the disease caused by the SARS-CoV-2 coronavirus. Half of my lab on the research side, we've been developing, one example, we've been developing new diagnostic tests that can be used to identify coronavirus infection. We've also been interested in doing a genomic surveillance, which is viral genome sequencing as a way to track in real time how the virus is spreading in our communities and across the world. And we've also been interested in identifying perhaps new ways of diagnosing the infection, perhaps by looking at host response sequencing as a way to look at the patient's immune response. On the clinical side, the main area of need right now for us is the ability to ramp up capacity for diagnostic testing for this virus. Unfortunately, the United States is behind the rest of the world with respect to being able to test every patient who needs to get tested. And it's already been clear that the coronavirus has been seen in the United States now in the majority of states in the United States, and it is circulating here. So it's absolutely imperative for us to be able to diagnose infection both rapidly and accurately as a way to prevent further spread, because we can put these patients who are identified with infection in isolation, be able to treat them as well as trace contacts.
Excellent. Well, thank you, Dr. Chiu. We actually have a polling feature now enabled on this town hall. So one poll that we're going to put up in just a second is to see how many people here on the town hall today. I see we have 84 people live joining us. How many of you are self-quarantining? I think that's a good measure of how worried you are about the spread in your own local communities. So we'll put up that poll in a second, and if folks can vote on that, that's going to be very interesting for the panelists and for our discussion today. And also, we're curious to see where you're joining from. So if you want to just type in the box and welcome and say good morning. I know there's a lot of people still eating their breakfast or in their pajamas. We picked this time so that we could be sure that we get the Dr. Chiu. So we picked a very early time. So we're seeing good people coming in from Chicago, New York City, Germany, San Diego, Cambridge, UK. Excellent. A lot in San Francisco as well. So Israel. So excellent. Well, we've got a global audience. So could you go a little bit more deeply, Dr. Chiu, into the genomic work that you're doing at UCSF? Because you mentioned how do we track the spread of the samples. Now that you've got a lot of samples coming in, you're able to test, but you're also able to sequence the genome of the viruses. What are you learning for that, and how could we use that in the future?
Yes, so a powerful tool that we now have in the setting of a public health outbreak is called viral genomic epidemiology. And the general idea behind it is that we take clinical samples from patients who are infected with, in this case, the coronavirus, and we sequence their genome directly from that sample. And what we can do is, as an outbreak is evolving, we can identify the mutations, the polymorphisms, mutations that this virus may accumulate as the outbreak is ongoing. The mutations can be viewed as similar to a DNA, RNA fingerprint, molecular fingerprint. And it's already been shown that, for instance, in other outbreaks, such as from Zika virus and Ebola virus, that we can actually use this genetic sequence as a way to track, for instance, how the virus is being transmitted, whether or not, for instance, all of the viruses in a certain cluster are all originated from a single strain, or whether we're seeing kind of different introductions of the virus into the United States from many different areas. So our focus has been on California, and we have been interested in doing genomic surveillance of the virus in California. To give you one example, just last week, I made publicly available, the fact that all of the sequences from the Grand Princess cruise ship, which was off the port of San Francisco, based on the sequencing, it appears that the strain that was circulating there on the cruise ship is the same strain that is circulating currently in Washington State. So it gives you an idea, perhaps, of how a virus can sort of spread over time, and also inform public health agencies of what we can do to sort of minimize spread of the virus.
Great, excellent. That's good to know. And I saw that you tweeted a couple of days ago the phylogenetic tree of those, so you can actually go online and look at Dr. Chiu's tweets and see that. We have the poll up, so if people want to start polling, I can see everybody's polling away, so that'll be great data to look at in a second. So a reminder, this is live Q&A, and if you want to ask Dr. Chiu a question or any of our panelists, just type your question into the Q&A box, and if you've got your microphone and video enabled, then we can actually dial you in and you can ask it live. I want to just go around quickly then to our other panelists and get a quick update. So Trevor, can you explain you came on to our show last week and you talked about the white paper that you did. This is the first example of CRISPR being used as a diagnosis for the new virus. And can you just explain then the update that you published yesterday and the significance of that preprint publication?
Yeah, so we were really lucky to work with Charles on the clinical side to actually kind of take this really new technology, which is CRISPR-based diagnostics, and very quickly, and as it turns out, very effectively, kind of move it into a situation where we're able to actually collect some data looking at the validity of the test. So the really important step in this preprint is like actually using it on real samples, because there's could be a huge difference between doing things in this kind of synthetic world and doing things in the real world. And you want to kind of get to that real world point as fast as possible, especially when you're making diagnostics. And I think in general, what we're really excited about and kind of our long-term vision is that we have a bunch of technologies that can be used just kind of exclusively in the lab, and there is a desperate need to scale those technologies so that more tests are available. But I think what also this kind of COVID-19 pandemic has shown is that there's this huge gap in just what's possible in diagnostics today. And that gap is the ability to have really accurate, really sensitive and specific diagnostics like what we have in a hospital setting or in a clinic, but in a decentralized manner. And really this paper is one of the steps in kind of getting from things that work in the lab to things that could work in an airport, in a field setting, even at home eventually, that are giving you the same kind of high quality result you get in the clinic, but in a more easy to use decentralized format.
Excellent. And Rebecca, if you could just explain the significance of what Twist is working on and the significance of the product that you released this morning.
Yeah, so Twist is committed to delivering tools to support the infectious disease community, and we're utilizing our core competency in high-throughput DNA synthesis at scale to support those efforts. So what we launched this morning are synthetic RNA controls that can be used as reference standards for the use and development, verification, and validation of diagnostic assays and monitoring the day-to-day assay variation. So these synthetic controls are from two strains that are circulating in the community. They're fully synthetic, so there's low biosafety concerns. They're sequence verified, and they can be used as controls for both RT, PCR, and NGS target enrichment-based assays.
Great, excellent. And Will, if you can explain the significance of what you're bringing to the table in terms of the hardware for the testing infrastructure.
Absolutely. Thanks, John, for making this happen. As many in this community have known, you've known Opentrons for a long time as the most affordable place to get your liquid handling robots. And until 10 days ago, we were really just focused on helping researchers who don't have any robots at all. About 12 days ago, I guess now, the emergency use authorization information came out, which meant that our tools could also be applied to this crisis in a meaningful way. And so since then, we've been really working over every waking hour to deliver a product that can actually do a lot of screening. And so right now we're installing the first 10 robot system down in Mountain View that can do 2,400 COVID-19 tests every day.
Excellent. Thank you, Will. And we'll come back and you're going to show a couple of pictures of the of what your hardware looks like later on. So going back to you, Charles, if you could just kind of, look at how this research community, how this industry is kind of reacting and providing these tools and services to the testing community and the clinical community. What's the significance of this and the three different services that are being provided here?
Yeah, well, I have to say that all three of these services have the potential of being absolutely critical in helping us to manage the outbreak. And as I said before, right now our main need is really diagnostics. It's diagnostics that drives both the development of vaccines and therapeutics and also informs the public health response. So the simple fact is that if we don't have diagnostics, we're not going to make much headway in terms of being able to address the COVID-19 outbreak. So just going from step to step, from Trevor's work that I've been in collaboration with, it's absolutely critical that we have very rapid diagnostics that can be moved from, say, the clinical lab to point of care settings. What he's defined as the emergency room, doctor's office, maybe at the airport, or even ultimately at home. Because one of the tools that we use to prevent infection, the only tool that we really have, may be self-quarantine, the ability to keep social distancing and keeping infected individuals away from each other as a way to prevent spread of the virus. So having the ability to test at home is going to be critical, and really moving or decentralizing testing is going to be important. With respect to Rebecca's work with Twist Biosciences, we absolutely need controls with all of the assays that we do. To give you an idea, it was very difficult from the early on for us as well as a community to develop diagnostic tests, because we simply had to work with infectious virus cultures. And so that was a hindrance, because in the United States for a very long time, actually several weeks, there was no access even to infectious viral controls. There was no one culturing the virus, no available source of the virus. So the ability to work with synthetic controls where we don't have safety concerns, I feel moving forward is going to be critical with being able to rapidly validate new diagnostic tests. And then the third is the development of essentially high throughput automation, the ability to go through hundreds, thousands, maybe even millions of samples. And pretty much for us, at least for the smaller academic laboratories, we're less experienced with the use of high throughput. I mean, we can get to 50 samples a day, 100 samples a day, maybe a couple of hundred, but we're not used to getting to the thousands of samples or the tens of thousands of samples a day that will likely be needed to address the outbreak.
Great. Trevor, Charles talked about the need for in-home testing. So from this paper that you published yesterday to the ability to have a colorimetric strip test, which is what your paper showed, just like a pregnancy test kit, you could actually have a strip test kit. So what do you need to get that into our homes?
Yeah, no, definitely it's a step along the path. It's not something that'll be available tomorrow, but it's a good, important kind of first validation that this is something that actually does have the sensitivity and specificity that you expect and need from something like this. I think a key thing from us is working with partners who do have experience in scaling these tests kind of both on the lab side, but then also in things like cartridges, and then of course eventually for kind of more at-home type and like in doctor's office type formats. So definitely we're very interested in speaking with companies that want to leverage this chemistry to kind of address this outbreak. And we're working with a lot of existing partners, but very, very open to new partners as well.
Got it. Charles, I contacted my local health department here in Contra Costa County to let them know we're having this town hall, to let them know that these new technologies are going to be coming online. How are we seeing the response? Both, let's just talk. We're all in California right now, so let's just talk about the California response. How are we seeing the state and the county level managing this, and what more do you think we could do and should do to engage and help the spread of the pandemic?
Yes, well, again, at the state and county level, as I said before, the key tool that we really have is quarantine or isolation of contacts. And so the recommendations have been for instance, if you feel that you are perhaps getting sick, I mean, there are a couple of common sense measures that you should immediately take. One would be that it's very important that you wear a mask if you feel that you're getting sick. Continue to have good hygiene, regular hand washing, avoid sneezing or coughing on someone else, etc. But at the same time, because of the concern that there may be spread not only within the community, but there may be spread within hospitals, or even, for instance, in the emergency room, many hospitals have set up essentially tents, including UCSF. We've actually set up respiratory or tents in front of the hospital where we could screen or triage patients who may have respiratory symptoms. Wow. And everything is being done in close coordination between hospitals who are identifying these patients, again, using the help of diagnostic technologies that are rapidly being ramped up. But the idea is that this is all done in close coordination with both county and state and federal public health authorities. So in other words, cases as they're being identified and diagnosed are promptly reported to public health agencies so that they can track these and monitor these patients as well as their close contacts. Because it's really working together in a kind of as a community is going to be the best way that we can minimize or mitigate the effect of the outbreak.
Great, thank you. So a reminder, this is a live town hall and we have two features to interact. We've had 16 people ask questions. I would like to ask the audience to help me decide which questions we should get to. So if you want to upvote the questions, just hit the little thumbs up and we're going to go to Carl Handelsman live, who has a question in a second. But before we do that, I want to give you the results of the poll. We asked, how worried are you about the coronavirus and its spread? 38% of you very worried, 48% of you somewhat worried, 14% of you not too worried, and nobody was not worried. So everybody on some level of the worriness scale. Are you quarantining? 21% of you said yes, you're self-quarantining. 48% said yes, workplace measures, and 39% said no. Wow, that seems pretty high. But again, we've got a global population here. So we're not sure how that breaks down geographically. And then the third question we asked was, what's the most important thing synthetic biologists can do to respond? And 85% of you said engage with other researchers on R&D solutions. 39% of you said engage with the local community and authorities, and 41% said engage with policymakers. So great, excellent. Well, thank you for doing that poll. And I'm just going to share the results with everybody now so that you can take a look at those specifically. And then we have a question from Stefan Breinlinger. So Stefan, I'm going to have you ask your question just right after Carl Handelsman. So Carl, can you hear us? Maybe you're muted, Carl. So we'll go to Stefan to ask your question. Stefan, you should be able to ask your question now.
Can you hear me?
We can, yes, go for it.
Yeah, so I'm very worried about false negatives of symptomless or mild patients or patients that are in early incubation period. How can we address this issue, especially if we apply those results to travel restrictions or to travel decisions? I'm really worried about the false negatives that we can get from this mild cases.
Charles, what are your thoughts there?
It's a great question. I can tell you that based on what we know of the trajectory of an infection, usually at the time of onset of symptoms, when a person first starts feeling sick, that's actually when it's actually easiest to identify the virus. What's interesting is that during the course of the infection, the severity of the illness does not appear to correlate with the amount of the virus that's found in, say, a nasal swab or another sample that you may be taking from the patient. And indeed, you're right to be concerned about false negatives because the viral loads can actually fluctuate over time. So I do think what it does, it really highlights that the importance of doing testing, of having not only the ability to do tests like one per patient, but the ability to do what we call serial testing, to actually continue to do testing on a regular basis to monitor these patients. Certainly, we already know that a single test does not exclude infection. And this is why the development of rapid kind of tests low-cost tests that you could do, for instance, multiple times a day, or at least multiple times per patient, is going to be really helpful in us, because we already know that a single test alone does not exclude infection. The other thing I want to say is that it's really important that we not only maintain Qpcr or reverse transcription polymerase chain reaction as our key test, we need to develop other kinds of tests to diagnose this infection or to track it. And namely, what's probably coming up the pipeline is a serological test, a test that looks at antibodies, because we'll have a better way of determining how prevalent the virus is in the community and how many people have been infected. Currently, without such an indirect test, we have no ability to track this.
Great, thank you. We have a couple of questions on current methods using Qpcr and then also about reagents. Carl Handelsman wants to know, is there a reagent shortage, RNA isolation columns, etc.? And John Oliver wants to know, how does Mammoth Diagnostic compare to the Qpcr test released by the CDC? So maybe, Charles, is there a reagent shortage? Going back to Carl's question.
Certainly there has been reported reagent shortages, and the critical reagent that we need are these extraction kits. So the reagents and materials that we use to convert a clinical sample into DNA and RNA. We're not personally experienced reagent shortage, but I think it's going to be imperative on manufacturers for these reagents that we develop, that we continue to maintain the pipeline, because one of the key steps is actually nucleic acid extraction. That being said, some of the technologies, such as some of the isothermal amplification and some of the CRISPR technologies that Trevor and at Mammoth has developed, and others, you might have the ability to actually bypass the nucleic acid extraction step. So it gives you the possibility, even though we're not doing that now, it's certainly a possibility, an option in case we simply don't have enough of these extraction reagents to test patients.
Trevor, Will, any comments from your side on reagents and methods?
Yeah, no, I think Charles is exactly right, that especially as the kind of CRISPR-based diagnostics further develop, one of the key advantages could be very simple sample prep, so you don't need to do this more complicated extraction. And that's definitely like an obvious kind of next step in that process. And I guess going to your second question, definitely in the kind of preprint we published, we do have initial sensitivity specificity data that's very encouraging, but there's further validation that needs to be done before you could say the kind of exact relationship of the CRISPR test to the RT-PCR test, for example.
Great. And then I guess for Will, specifically, we'd like to know, what's the real limiting step in nucleic acid testing? Will there be enough people to do the sample pre-treatment and the RT-PCR? Can you answer that, Will?
Well, definitely the purification is the longest part of the process, and where automation can really help scale higher throughput for the smaller labs. But it's really interesting to hear that some type of crude lysis could be a possibility, because that would certainly shorten the process and enable even more labs to do this. I think that having a diversity of tests also lends the weight on supply chains. And so that could be a real key advantage as well. I will say that our reagent partner, who manufactures all of their magnetic beads in the United States, is right now creating 150,000 test kits per 2 days. So their manufacturers are definitely ramping up. And if you know where to look, these things can still be found.
Great, excellent. Charles, I'd like to know from you, what tools, technologies, hardware, software, or other resources do you need right now from our community to help you do your work?
Well, I think it's really kind of what we've been discussing, the development of these broad-based diagnostic tools as a way to diagnose COVID-19 infection. I do have to also mention that another need, which I think is going to be critical, right now, understandably, the need is for better diagnostics for the coronavirus. However, very shortly, it's going to be probably a situation where you have patients who come in. Most of the time, even now, the coronavirus diagnosis is actually still quite uncommon, which is good. Hopefully, we can keep it that way. However, there may come a time in the near future where a patient who comes in or an individual who comes in with respiratory symptoms could have equal probability of having coronavirus infection, but having been infected by perhaps another respiratory virus such as flu. So the ability to develop multiplex tests that would test not only for coronaviruses, but other respiratory viruses and other respiratory pathogens simultaneously is going to be tremendously effective. To give you some idea of how how inefficient our testing is, we have separate tests that we use to diagnose COVID-19, and we have another test that we use to broadly diagnose other respiratory viruses. What we really need is a multiplex test that can essentially diagnose all respiratory viruses in a single test, and that way will also help with limiting reagents and limiting resources.
Great, excellent. And I want to ask from a practical perspective, when the virus actually, let's say that I get an infection in my nose, how quickly does that propagate, and where does that propagate to throughout my body? And then how many copies do you need to be able to, for a test to be able to pick it up, and how sensitive is that test in the timeline of the infection?
Yes, so the incubation period of this virus is thought to range from anywhere from 2 to 14 days. And this is why we have a 14-day quarantine or a 14-day self-quarantine for individuals who may have been exposed. On average, the median time of incubation is about 5 days, meaning that 5 days after, on average, after you get exposed, you may start to develop symptoms. As I said before, this virus is a little unusual. It's more like influenza in that respect than it is to SARS coronavirus, in that you're apparently most infectious. You have the highest viral loads at the time that you have symptoms. And this was unlike the case in SARS coronavirus, which happened in the early 2000s. With SARS coronavirus, as you became sicker and sicker, that's when you had higher and higher viral loads. It appears to be different for this virus. And that's why it's really important for us to be able to continue to test patients during the course of their hospitalization. In some ways, it's not enough to simply just test once, make a diagnosis, are you infected or not, and then you're done with diagnostic testing. In fact, another critical aspect of testing is, when is it safe to discharge patients from the hospital? And this is why you're typically for patients who have this infection, who are hospitalized, they actually get multiple tests for COVID-19, as well as tests for other pathogens that may be causing secondary infections during the course of their hospitalization, which really reflects the importance of better diagnostics. The other thing I should mention in terms of what is needed is that we critically need these tools, ways of using synthetic biology tools to provide targets or to identify targets that could be used in the generation of vaccines. There's a lot of interest right now in the development of DNA-based vaccines, which can sort of overcome many of the limitations of growing essentially vaccine candidates in cells. So the ability to rapidly synthesize and target parts of the virus will enable us to hopefully get effective vaccine candidates out there very quickly, because ultimately I and many others feel that the vaccine may be the only way that we can actually eradicate this virus from the population.
Great, thank you very much. And that's a good time for me to mention the next 2 town halls that we're planning. We're getting into a weekly cadence now, and we have 2 coming up. One will be on environmental sequencing of the virus in the environment. We're going to sequence the BART. We're going to sequence the New York subway. That's some work that that Twist is doing, and to start looking at how we can look at the spread of the virus before it gets into human contact. So we'll do a special town hall on that coming up, and another town hall that we're going to plan on synthetic biology tools and technologies for vaccine development. There's a lot of exciting work going on in that area, so we're going to focus on that in the next 2 weeks. If you want to find out when those are going to be, then sign up to our newsletter at SynBioBeta.com. Dr. Chiu, I said we'd only take 30 minutes of your time. You're extremely busy, and we prefer you out in the back in the lab and helping these poor patients who have coronavirus. So thank you very much for joining us this morning. We really appreciate it.
Thank you so much.
Thank you. So we're going to continue the panel discussion, and I want to remind everybody that we have live Q&A. So if you have a question for the panelists, then you can upvote them and downvote them. We're also going to be asking a few new polls, and if you have suggestions of polls that you want to know from the audience, we have 116 people live online right now. Then tell us which polls we should be asking. You can put that in the chat box. And I'd like to do a poll to find out when is the best time to host these weekly town halls. So would it be 7:30 a.m. on a weekday? Would it be no, Rebecca, we'll not do that. Would it be 8 a.m. on a weekday, 9 a.m. on a weekday, or should we look at early morning on a weekend, like 9 a.m. on a weekend, which is when we did the first one last week, which was very popular as well. So while we tee up that poll, I've got a few more questions. One from Ted Anton, who says, why do these viruses make more of a threat in winter than in summer? Actually, that would have been a good question for Dr. Chiu, but we ran out of time. But if any of the panelists want to take that, then we can take that. And then Josh Shirnoff has a question. Why do we see so many different assay designs emerging, particularly Qpcr? And is there convergence on one in the healthcare system? So does anybody want to take the winter question, or anybody want to take the why have we got so many different assays?
Take the assay question.
Sure.
Again, and I'm sure others can elaborate. But
yeah.
I think yeah, it definitely can be confusing that there's all these different techniques out there. And even for like RT-PCR tests, there's, there's the CDC test, there's the WHO test. And I think there are huge advantages to having a single test, because then you can standardize and it's a little bit easier to normalize to what you expect versus what things are surprising. That being said, though, I think especially as we start thinking about decentralizing testing, and also as we think about what Will brought up, which I think is really interesting as well, which is the decentralized supply chain. So you're not relying on just like one reagent for one test that can bottleneck the entire process. And different assays can have different strengths and weaknesses. So for example, the serological test, that can tell you if you maybe previously had an infection, but maybe it could be less sensitive for if you have an infection now, whereas the RT-PCR test won't tell you if you had an infection previously, but is extremely sensitive ideally in telling you if you currently have an infection. So there's kind of different goals for different tests. And then layered on top of that with things like CRISPR-based diagnostics, we're really kind of thinking about how can we move these things outside of the lab and make them simpler eventually. So there's all these different goals for the tests. And then even within a particular type of test, I think diversifying the supply chain and diversifying the type of testing so you could hit it from different angles and validate these tests is pretty important.
Excellent. Rebecca, did you have any comments on that?
No, I agree with that 100%. Absolutely.
Okay. We've got a few questions on the positive control that you just released, but we'll come back to that in a second. Will, do you mind just sharing your screen and just bringing up a picture of this hardware system that you're setting up in Mountain View? And then if you could also explain about these emergency licenses that have been granted and what that means. And then we have also a question from one of our participants on the need. Do you see a need for CLIA certified labs? So if you could take that on as well.
Sure.
And yeah, maybe we don't have time for the full presentation, but maybe just back to a few of the key.
I just wanted to bring up, this is just the process that most labs are running, the RT-qPCR. And Opentrons can really help on the sample plating, RNA extraction, and the plate setup portion. The Qpcr will be your Qpcr instrument. And we've broken it into 3 different workstations. The first one is when the collection tubes comes in, the nasal or oral swabs, it will be in a tube with buffer. That needs to get put into an extraction plate. This station should be in a separate room because the uncapping of all of these tubes is a pretty contamination frenzy type of process. And so separating it from all the other lab work is very important. So then you'll take that extraction plate and go put it on station B for the RNA extraction with magnetic beads. This process takes 3 hours because there's a lot of wash steps to make that the RNA really pure. So that's definitely the bulk of the system. Then the next step is go into your Qpcr room, which should be your cleanest room, where the robot will mix the RNA sample with your Qpcr reagents. Then the plate will be ready to put on your Qpcr. So we're actually suggesting an 8 robot system for the highest possible throughput while still maintaining sort of operational efficiency. You could obviously scale this down from an 8-robot system to a 4-robot system, or you could scale this up from one 8-robot system to 2 8-robot systems and beyond. But with this system, you can do 12 plates in 12 hours. So that's roughly 2,400 samples per day. You've got 2 of the reformatting robots leading the charge, 4 of the extraction robots there in the middle, and then 2 of the Qpcr robots. And so this is roughly how the timing of that process will break down. This is only the sample prep, not your Qpcr time, which can be a bulk of the time. But just noting that this timeline does not account for that. So you've got the 2 A robots kicking off the first 2 B robots, which take 3 hours to do their extraction. Meanwhile, the first A and B, the first 2 A robots, excuse me, are kicking off the 3rd and 4th B robots. So basically this shows that you can run the extraction robots 3 times in a 12-hour period, and the rest of the robots sort of flow around that. So that's the overall system.
And Will, who's your target market? If people are looking at this and they want to get in touch with you, maybe explain how they do that. But where are you seeing this kind of system particularly fit?
Absolutely. So with the emergency use authorization, any CLIA lab with a BSL-3 capacity can be running these types of tests. Much of the testing bandwidth that's going to come online will be the Quests and the LabCorps of the world, the labs that are already highly automated, which is awesome. We need that bandwidth. Where OpenTrons can be most effective is taking labs from 0 automated bandwidth up to this 24 samples per day within a week of your order.
2,400, right?
Yeah, 2,400.
Yeah.
Excuse me.
Okay.
And the real advantage of the OpenTron system, it's a lot of small robots instead of a few big robots. But if you only have 1 robot for each of these steps A, B, C, if one of those robots goes down, your entire platform has stopped production. So having lots of smaller robots improves redundancy and therefore resilience. The other thing is that these robots come in a FedEx box, not on a forklift. Then just the final thing that I'll say is that this process is not breaking new technical ground for OpenTrons. We're doing sample plating, RNA extraction, and Qpcr setup in hundreds of labs around the world. And this is really just stringing this workflow together to provide a high throughput system of work that we're already doing in other labs.
Excellent. Thank you very much, Will. I appreciate it. If you want to unshare your screen, we'll go back to the other panelists. And we have a lot of people who have questions. So if you want to vote the questions up or down, those are the ones that I will prioritize. Tom Knight, if you're connected to a mic and you want to ask your question live, then just put your hand up and I'd like to come to you to talk about reference standards. And if anybody else would like to ask their question live, then just put your hand up in the Zoom app and I will call on you. Particularly, we also want to remind you that we're going to be doing some polls. So if you have ideas for polls that you want to ask the audience, then we can come to that as well. Okay, so we have somebody wanting to ask their question live. That's David Kong. Yeah, David, welcome.
Hello, everyone. John, thanks so much for organizing this really important gathering.
Sure. And if you just want to introduce yourself and say where you're from, David.
Yeah, sorry. My name is David Kong. I'm a director of the Community Biotechnology Initiative at the Media Lab at MIT. So my question is actually a little bit more about public engagement and basically spreading knowledge and education about what's going on. There is an incredible deficit of knowledge in the public right now. One thing I've personally started doing the past couple of nights is just doing Instagram lives, Facebook lives with just my local community, which has been really effective at getting people to understand more what's happening. I'm fortunate to have relationships with folks like this guy, Jaylen Brown from the Boston Celtics. I got him to do something. He did an Instagram Instagram live last night. We're doing one together tonight. I'm also connecting with networks, folks in Hollywood, et cetera. One thing I'm hoping to try to organize is like a pairing of things like influencers plus technical people, scientists, public health officials, et cetera. We're getting right now, the communication from the administration, from officials is so low. So getting these types of things organized like ASAP, I think feels urgent. If any of you are good science communicators or know folks that might be effective at this, let me know. And also I would encourage you, so it's also a question for everybody, if you'd be willing to potentially start taking on some of this communication process in addition to the research. Obviously, we're all doing things in labs as well, but this is another kind of major moment right now where people need to know. So
excellent. Thank you, David. Really, really good suggestion. I'm glad that you're taking on that initiative and doing that in your local community. So thank you. I want to just, we can come back to the panelists for a second on outreach ideas, but I also want to come to Tom Knight's question. He says, I would like more information about the twist positive controls. Is it full length? Are there concerns about the biosafety of the positive controls? So Rebecca, if you could give us, if you could just remind everybody, for those who joined late, what Twist has released this morning, what the positive controls are for, and an answer there to Tom's questions.
Right, so what we talked about earlier today was Twist Bioscience is releasing fully synthetic RNA controls to enable the work in the infectious disease community. These are fully synthetic. They're not originated from human samples. It's a BSL level 1. What we've used is the high throughput DNA synthesis capabilities of Twist to generate DNA and from that make RNA. So it's full viral coverage, and we've validated that in a variety of ways through next generation sequencing. We've also used it as a positive control for one of our research use only RUI and GS target enrichment panels that we've designed specifically for SARS-CoV-2, which we are also releasing for distribution early next week, along with these synthetic standards. Excellent,
great. The next question we have from Khalid Alam, who he's on a desktop without a mic. He says, CRISPR-based diagnostics, are the licenses from UC Berkeley and Harvard, MIT Broad exclusive? Are you limited to use by Mammoth Sherlock, or are they applying the inclusive innovation model to the technologies? Trevor.
Yes, that's a great question. I think at Mammoth, something that's really important to us is working with researchers and people that want to expand on the technology. That's why we've been extremely open about everything we're doing. So we published a white paper very quickly. Now we've published this preprint. Of course, hopefully journal article coming out soon. And we're dedicated to continuing that transparency and working with people that want to build on it. I mean, I think I agree with the philosophy around this type of technology will only be successful if people are open about what's going on and have the ability for others to build on the work that's been done. So it's a classic kind of shoulders of giants situation. And we're very interested in working with anyone who thinks they can use or develop the technology, for sure.
Great, excellent. And Trevor, if you could just go into detail, we were speaking last night about the paper and about what this kind of testing that you've developed will enable. It's not just for one specific virus, but it really allows you to do a whole panel and not just viruses either. So if you could just go into a bit more detail, maybe about the vision for Mammoth Biosciences and where you see this going in the future.
Yeah, so the vision is definitely that I think this particular pandemic has exposed a huge gap in just the molecular diagnostics industry in general, where we don't have a way of doing really accurate, really fast, really easy to use molecular testing, just kind of in a decentralized fashion. And I think the long-term vision is that using, for example, CRISPR-based technologies, you could develop a diagnostic test that's in the format of something like a pregnancy test, but is giving you results as if you'd run a PCR test in a lab. And that's obviously a super high bar, and that's why the first steps are, like we've published in this paper, doing things in a lab, validating that it's actually got the sensitivity and specificity that you think it should have. But what's exciting is that even in that lab format, we're using things like lateral flow strips. And that removes, for example, a really expensive piece of equipment, which is some sort of optics that's actually going to read out, say, a fluorescent signal or something like that. Instead, you can actually just look at the lateral flow strip with your eye, which is a really great optic as well, as it turns out. And you can actually just read out, okay, is there a positive or negative? And like we mentioned a little bit before, there's also benefits on the front end. So what if you don't need to do these really kind of complicated sample prep steps, which obviously systems like Opentrons are like enabling labs on some level to do any of this. But hopefully you could simplify that so it's not such a drawn out process. And I think right now the advantage is that even in a lab setting, you're able to decrease the time it takes to actually get to these results, and you're able to potentially expand this testing to labs that don't have the fanciest equipment. And also you're able to expand it to labs that on the other end of the spectrum don't necessarily need to run thousands of tests. What if they just want to run 1 or 2 tests early in the process, or like in a field setting or things like that? That's kind of where the technology is starting to enable things. And on top of that, what's great is that you're getting, ideally, the sensitivity and specificity that you'd get from just like the classic techniques. So you're not sacrificing that.
Great, excellent. And yeah, Rebecca.
Yeah, if I can build on that, I think what Mammoth is doing is really exciting. Paper-based assays are a fantastic way of getting these tools into the community, into more hands. But one thing that Charles Chiu talked about was a way to diagnose not only one virus at a time, but be able to look at a panel of diseases with a single test. And so what we've done at Twist is we've been committed to, like I said, supporting the infectious disease community for many years now. We started work with Illumina on our Pan-Viral 1.0 panel. We were able to detect Ebola from West Africa, monkeypox from Nigeria. Again, we're developing research-use-only tests right now to not only detect SARS-CoV-2, but we're also developing panels for coronavirus families. So if you test one patient to understand what coronavirus they may have, or in order to track the disease and how it migrates over time and how it evolves. We're also developing, again, for researchers only, a respiratory panel. So if someone does walk in and they have a symptom, then you can use one test to diagnose if it's a variety of diseases. So this is something that we're really excited about and working to develop in our R&D team.
Great. And can you give us a teaser, Rebecca, of the future town hall? Emily LaProust is going to join us to talk about environmental sample sequencing. Or maybe that's somebody else at Twist.
That's probably somebody else at Twist. I'll let Emily talk about that.
Okay, no worries. Fernanda Gandara says, can you reconcile the headline of IDT shipping 700,000 tests early in the week and all the public labs now testing? Quest and LabCorp started testing on Monday with the still low testing numbers this week and current shortage of tests. What's the current bottlenecks? What will be the future bottlenecks? And Carl Schmieder said he'd heard that IDT is already at capacity. It's probably not a great idea to rely on one company to create and ship tests. Trevor, any thoughts on that?
Yeah, so I mean, obviously I don't have any particular insight into IDT, but I think it goes back to the question we talked about earlier, which is just another reason for the importance of the diversity of tests. You're not relying on one supplier at any part of the chain. And I think obviously a bunch of labs right now are spinning up their testing. So there's always some lag time between reagents being available and validated tests being available. So that could be part of it as well.
Great. I'm going to come to 3 call-ins. Mickey McManus, I'm going to have you go live shortly, and then I'll come to Isaac Larkin, and then Stefan Breinlinger. I see you've got a lot of insight there, so I'd like to come back to you, and maybe you could introduce yourself and give us your insight. And just a reminder, this is live Q&A, so if you've got questions, type them into the box, or you can upvote them, and I'll ask the best ones. And it's great to see everybody engaging and answering each other's questions in the Q&A panel. So if you haven't opened that up to look at it, then I encourage you to do so. So Mickey, if you want to just introduce yourself and go ahead and ask your question.
Hi, John, thanks. Mickey McManus, I'm a senior advisor at Boston Consulting Group and fellow emeritus at Autodesk. And I had a question about maybe what Mammoth Technologies is doing, or Mammoth Bioscience. Trevor, on the standing of what is the scaling implication, or what is the bottleneck for scaling what you're doing with CRISPR-based tests? Like what's standing in the way, or what would stand in the way in the next few weeks or months?
Yeah, so definitely in terms of the tests that we outlined in the paper, there's a big need for just working with partners that have huge manufacturing capability for each of the components. Anything from like lateral flow strips all the way through to obviously just the reagent chemistry itself and the proteins. So that's one part of it, I think, and that's like the short-term scaling. I think on the long term, it really comes down to working with partners that, for example, have cartridge-based technologies that this chemistry could be put on to enable this type of testing in that kind of more decentralized format as well. And then on the kind of long-term scale, it really comes down to integrating it all into a disposable, very easy to use type system that is manufacturable. And I think a lot of kind of decentralized diagnostics technologies have fallen down a bit on the last one on the manufacturability of it. And that's where working really closely with individuals and partners that have a lot of experience in manufacturing at scale, like hundreds of thousands to millions of units, is really important from the very early stages.
Great, excellent. Isaac, over to you. Maybe Isaac doesn't have a microphone. Stefan, can we come back to you? It sounds like I'm guessing you might be in Germany, but maybe you could introduce yourself and ask your question.
Yes, thanks again for and also for organizing this this panel. Yes, I'm from Germany. I'm a pharmaceutical biologist, researcher, and yes, I'm kind of worried about the situation, especially about the lack of taking it seriously from early on, especially from our leaders and politicians. And now we are paying the price. I think if you look at the official numbers, I posted one nice short read about the statistics of the official numbers that we have from early outbreak in China. And yeah, so one question I have.
And Stefan, sorry to interrupt you. Are you in Germany or are you in the US?
I'm in Germany.
Okay.
Right now,
and do you mind saying which institution or company you're with?
Yes, I'm working with the Martin Luther University in Halle, which is like in eastern part of Germany, close to Berlin. And yeah, so it's an Institute of Pharmacy, Pharmaceutical Biology, and yes, so
Great.
I'm
Welcome.
actually a natural product researcher. So
excellent.
Yeah.
Well, why don't you drop me an email, because I actually there's somebody interested in a natural products grant for the Gates Foundation award that they just announced, and might be interested in collaborating with you. So john.cumbers@SynBioBeta.com. I'd love to connect you around natural products. But anyway, back to your question.
Thanks. I will do that. Well, my question is there any reliable data about the virus surviving higher UV radiation and especially higher ambient temperatures, lower moisture content in the atmosphere? So is an early spring maybe helping us fighting this pandemic? Has there been any investigations about that? Because I think prolonging or flattening the curve of new infections could really help us in fighting this pandemic. So I'm actually not seeing a vaccine or more testing possibilities as something that we can accomplish in the short time that really we have to because I think that all the new testing kits and testing possibilities, I think we will not be in time to really fight this pandemic through that.
I'm not sure.
The next one.
Yeah, we don't have any researchers left on the call as Charles has left, but does anybody have any insight there?
I mean, my one thought, sorry, one sec, is when it's warmer in the northern hemisphere, it's colder in the southern hemisphere. And the reason I think that Charles and others say that a vaccine is really our only long-term way out of this is because this could become completely endemic in the global population long-term. And so I think, I don't disagree that we're a little bit late on the go for getting testing bandwidth to where it needs to be, getting a new vaccine and things like that. But better late than never, especially with the possibility of it truly becoming endemic in a global way. I also have been talking to people in the Southern Hemisphere who see this coming, and they need to be scaled up and ready to go when their flu season comes around.
Yeah, and just like Rebecca mentioned before about multiplex testing, you can definitely imagine a future where now flu season is flu A, B, C, right? A, B, COVID. It's not that hard to imagine. It doesn't mean it's necessarily going to happen, but like you said, there's, a range of different temperatures across the earth. So I don't think you can just rely on that. And I think what's important there is that I think if we rely too much on that, we don't, rely enough on the things that are actually quite simple. It's like, personally, it's been really eye-opening to me, like such simple measures we can take, like washing our hands, distancing ourselves, just doing things over Zoom can actually have huge exponential effects on spread. And I don't know, maybe that's just a general learning for the population for flu season in general. So.
Yeah, I think as a society, we've rediscovered the power of soap.
It's true. Anyone with kids knows that for sure. We have somebody asking a question about the viral sequences not being deposited in the public anymore, and I presume that's a biosecurity concern. I hadn't heard about that. If any of the panelists have comments on that. Others about what are the regulatory hurdles that will delay any of this technology, if there are any. And
I can maybe just talk briefly on that. I think that
sure.
The regulatory hurdles in the last week have become no longer hurdles and instead facilitators. I think that though being a little bit late, the federal regulatory infrastructure is now approving emergency use authorizations within 3 days of submission and doing a great job of making really clear what the requirements are. So I think while it was a little slow on the uptake, I think we're at kind of a good cruising capacity now to really scale up bandwidth day over day.
Great, excellent. Bill Slavin says, what CDC contacts or other government agencies can be reached out to for organizing local support for these efforts? The more direct, the better. And there's a whole discussion going on around the CDC primers being problematic. Any thoughts on connections to the CDC? Trevor, yeah, go ahead, Will.
Well, the CDC primers have been problematic. That's one of the reasons why this EUA was issued in the beginning, is they were seeing a lot of mismatch rates. And, you know, it's not entirely just, it's like the CDC is amazing scientists. It's not all their fault. There's an iteration 1 and an iteration 2 that happens in these processes. But now we're starting to see more diagnoses come on that are in the sort of iteration 2 level of primers, and they're showing way lower mismatch rates, targeting more parts of the genome, and are much better assays. So I do think that in the coming weeks, we're going to see a much better Qpcr test, as well as whatever novel technology tests come online.
Yeah, and it also really speaks to the importance of the controls. So like what we've done at Twist is we've created synthetic controls that you can use, synthetic RNA controls that you can use for your RT-based assays. We have 2 on the shelf, and we're also able to manufacture novel strains or novel sequences within 2 weeks. So other variants of the viruses can be made for additional testing for additional controls.
And Rebecca, are you the first to have that out? The first company to have a control out?
There are other synthetic controls that exist. The CDC recommends researchers go to BEI resources. ETC has controls in globally. I think the WHO recommends, I'm going to butcher this name, the Bernhardt-Nacht Institute in Hamburg. These synthetic controls, from my understanding, is that they don't cover the full genome. And this is one of the key differentiators that the Twist products has, is that we really cover the full viral genome.
Great, excellent. A question from Radiska Sanka. Right now, it seems like the supply chain is completely in a disarray and the test cure creation pipelines are too slow. How can we make this better? Is there a policy angle we need to work on? Any thoughts from anybody on policy? Yeah,
I definitely think long term, we need to see federal investment in this type of infrastructure. I think that, you know, this community has been doing a lot of work bringing sort of biosecurity and biomanufacturing to the forefront of national regulatory attention. But I think that this type of pandemic response has not received enough attention or federal grants as required. And I certainly think that over the medium and long term, having the government provide resources for doing testing and surveillance is going to be very, very important.
Great. And
I also have to jump off. There's a lab in India that's trying to deploy our system that I have to talk with right now. But this has been really exciting to talk with everybody. I really appreciate how people are really rallying to this call. This is something that we can all help with, and everybody's looking for their way to help. And it's really an amazing thing to see. So thank you all for joining. And I look forward to collaborating with you all as we continue forward.
Excellent. Thank you, Will. Thank you for the hard work that you and OpenTrons are doing as well.
Thanks,
we have
John.
a few last questions that we're going to wrap up, and then we're just going to bring up a poll to ask when is the best time to host these town halls in the future. As I said, we have 2 town halls coming up, one on vaccine development and synthetic biology, and the other one on environmental genomic sequencing. So that poll is going to pop up right now. We'd love to get your opinion on when is the best time of day to be doing these. Last few questions before we wrap up. We have Gagana Vandova. It is great that there are so many diagnostics being available. However, I was wondering how long would it take to get any of them into a clinical setting? Is it still the case that now the only people that would be tested are the ones which are the ones which have been traveling to a high-risk country or have been in close contact with a confirmed case? We are way past that being effective. What would patients with very mild symptoms be possible to be tested? Trevor, I guess over to you for that one. And we've answered some of those pieces as we go along.
Yeah, it's a really good question. And definitely the part around like who's getting tested is more of a policy choice for clinicians themselves. And I think that, yeah, that situation is changing really rapidly in terms of probably what the right approach is. But I think on the diagnostic side, I think what's kind of sad at the moment is that even people that do fall in these at-risk groups, from what I can tell, are having trouble getting access to testing. So it is really important that the FDA has done things like this emergency use authorization. And I think, like was mentioned before, these emergency use authorizations seem to be rather rapid, and it really has, I think, struck a great balance between, on the one hand, you don't want to put testing out there that isn't valid and isn't super rigorous, but on the other hand, you need to get testing available on the right timescale so that it's actually useful. And I think actually today we're in a good place there. Whether we could have gotten there sooner or not is a good question, though.
Excellent. We've come to the end of the town hall. It looks like we'll be doing them at 8 a.m. Pacific weekdays. That's 60% of people have voted for that. So thank you all for voting. Thank you to all of our panelists. That's Dr. Charles Chiu from UCSF, Will Canine from Opentrons, Rebecca Nugent from Twist Bioscience, and Trevor Martin from Mammoth Bioscience. So thank you all for joining. And this was a fantastic interaction, and we really enjoyed the Q&A part, and we look forward to doing one of these in the future. You can sign up to the newsletter if you're not already on there at synbio.com, and we look forward to seeing you next time. So thank you all for joining, and thanks to our panelists.
Yeah, thank you very much.
Cheers. Bye-bye now.